Here we go.
Alrighty.
What happened? You're alive. Welcome.
Good afternoon, everybody. Dr. Daniel Barth here.
I don't know. The system is all broken today.
Yeah. Well, this is our first show after April Fool's Day, right? So there you go.
Well, I made a fool out of me, didn't it?
And as usual, we're coming to you today courtesy of the book Star Mentor. So if you have a young astronomer in your life or someone who's just starting out, please go ahead and pick up a copy. If you have a copy and if you've read it, we deeply appreciate reviews on Amazon or other sources because we want to know what you're thinking. Let's go ahead and talk about today's episode. There's so much fun stuff in the space exploration news. The Artemis crew is being announced. We'll know. And the news that I heard, the announcer was breathlessly excited. The first astronauts in 50 years to return to the moon. And I'm like, yes, but that's not our national pride. That's kind of a national badge of shame. I think that we've been gone from the human exploration of space for half a century. It's just ridiculous. But I'm glad we're back. Yay, hooray. Take nothing off the engineers and astronauts and folks who are going out and doing those explorations today. Good on you. And I will be cheering for you. Yes, of course I will. Anyway, so we may have a whole program on that. But today's program caught my eye, and Paul and I, my producer Paul, were talking about headlines just before the program went live. And we talked about the idea of what's a good headline. Well, the headline that caught me was life discovered on nearby planet. And I'm like, okay, this is probably clickbait, but I'm Curious George on the internet. So I went ahead and clicked on it. And it was an interesting article. And actually, we're talking about an entire ecosystem, that an entire planet's worth of biology, quite literally. An entire planet's worth of biology. And it occupies a vast volume of space on a planet that no human being has ever set foot on. And it's not that far away. It's only about 500 meters away. That's something on the order of a little less than 2,000 feet. It's half a mile. If it's half a mile away, why haven't we ever been there? Well, it's half a mile straight down into the interior of the Earth is where this life lies. This deep biosphere is the term for it today. The deep biosphere exists, and we believe it extends down to several kilometers below the surface of the Earth. It likely, my prediction from the cheap seats, I'll bet it goes down a lot farther than that. I'll bet it goes down quite a ways. But they think it probably goes somewhere between 5 and 10 kilometers down below the surface of the Earth. And this layer of life is thought to be likely completely biologically distinct from the life on Earth. And that's an astonishing statement. One of the interesting things when you talk about DNA and is life on Earth all from Earth, and we see weird headlines like that every once in a while. Octopus could be from another planet. Well, no, they're not. They're cephalopods. There's lots of them. But if you take the DNA from any life form on Earth, it's very clear that it's one of us, that it's from here. You share a fair amount of DNA with a cockroach and even a tree or a bit of moss in your garden. You share quite a bit of DNA, certainly plenty enough that we can say, yes, all y'all from the same planet. So, what's interesting is we're talking about thinking that this life, this biome, excuse me, deep below the surface, could in fact be biologically distinct. And for me, an old time biologist, I'm looking at this and going, wow, this is awesome. The idea that you could say, oh, these life forms, these biospheres have been separated for billions of years. That's a fascinating idea because you could be looking at, oh, we can take a look at samples and we can test them and we can go, aha, this is what it looks like when we've got something completely alien to our biology, something that's completely biologically distinct and different from us. Y'all ain't from around here, are you? Yeah, that sort of thing. Because if we ever find life on another planet, on Mars or on one of the moons of Jupiter or Saturn, and we sequence its DNA, well, then there's the question. Are we going to find DNA that says, this is something completely different? Look, we've sequenced DNA from bacteria we found in an aquifer on Mars. And here we go. Would this hypothetical DNA sequence show something that's completely separated from earthly life forms? Or would it be perhaps 10 or 15% related, one of those ancestry DNA type charts that people are so fond of? Oh, you're 15% margin today. And if these life forms are showing DNA that says, hey, we're related, we may not have had any DNA exchange for, 2.5, 3 billion years, but we're related. We're related. And so this, looking at the biome of the deep biosphere and sequencing some DNA there would be a fascinating thing. This idea of early life is not a brand new one, friends. It is not. People began to suspect that there might be bacterial life deep beneath the earth in the 1920s, which is when we started mining petroleum, liquid petroleum, crude oil extensively. People have known for millennia that there are fossils in coal, and people have known about coal and coal seams for many thousands of years. And people have been burning coal, and you know that every once in a while, if you fracture a piece of coal, you see inside a fossil of a leaf or a sea creature, an anemone, something like that. While cool, life in coal had never been detected. And now this fellow, Charles Lippmann, UC Berkeley, he managed to culture bacteria out of freshly mined coal. So he took coal samples, freshly mined coal samples. Get your freshly mined coal samples here. It sounds kind of comedic. He took these freshly mined coal samples and he crushed them. And the coal samples were then sterilized at 160 centigrade for up to 50 hours before being crushed. So the idea was we want to take these samples and we want to completely sterilize the exterior. We want to completely sterilize the exterior, and then we want to crush it and see if there's any bacteria that emerge after crushing that would presumably have been alive in the interior. And when we think about these sorts of things, and there was questions of bacterial and fungal life inside crude oil samples. Were these indeed bacteria and life forms from the Earth's surface that had contaminated that oil as it came up through the ground? Or was this life that existed and lived in and perhaps fed upon the oil deep beneath the earth? And so the question was, how do we get a sample? If we're digging with a drill and we're drilling down through dirt and rock with a drill, how do we indeed get a clean sample that isn't contaminated by the drill and the crushed crap and the lubricating fluid, oftentimes water, sometimes mixed with other things, and it's slooshing around the outside of the drill to keep it lubricated so it doesn't overheat? And how do you get a clean sample out? Well, this was a problem, and it wasn't solved until the 1980s. And you're like, wait a minute, if they were looking at samples of deep life in 1920, and then indeed we're talking about a clean sample technology isn't developed until 60 years later, what's going on? It turns out there's another piece of that story, and we'll get to that in just a minute. So when we go and we recover samples from down there, what is down there? If we go down into very deep mines where people look for gemstones and diamonds several kilometers beneath the surface of the earth, and they find life down there, what are we looking at? What kind of life? Well, we have a large population of Archaea. And yes, does that sound like archaic, like ancient? Yes, exactly. These are the most primitive single-celled life forms, and they're thought to be amongst the oldest living life forms and species anywhere on the earth. These things tend to live in extreme environments. So they live in salt brines like salt lakes and salt flats, which are wet occasionally. They live in these brines. They live in acid pools where volcanic vents bubble through water, and the sulfur dioxide and carbon dioxide turn to carbonic and sulfuric acid and make for a very acid pool indeed. These things live in these acid pools. They live in hot springs at extreme temperatures, and you can even find them lurking around volcanic vents on the sea floor, thousands of feet down, complete darkness, no oxygen, and these bacteria live there. They're prokaryotic, which means they have no nucleus. They have no functional nucleus, and they're adapted for what's called a reducing environment instead of an oxidizing one. So imagine a atmosphere of carbon dioxide and methane and nitrogen, as opposed to an oxidizing atmosphere full of oxygen. So these are anaerobic organisms for the most part. Surprise, they're living kilometers below the surface of the earth, and they don't rely on oxygen. Well, what do you know? And so how do they get along? Many of these are what are known as sulfur breathers, which sounds very odd. And they metabolize, in other words, they're eating things like hydrogen, methane, ammonia, and they use metabolic pathways which don't involve oxygen. They are not evolved to live in an oxygenating environment. And they're inhaling or ingesting things like nitrates, sulfates, manganese and iron oxides, carbon oxides. All of these are electron acceptors, and basically that supports their metabolism. So these archaea are strange beasts indeed. Bacteria. Yes, we have bacteria similar in most ways to surface populations. Single cells, they have nuclei. They have common respiratory and metabolic pathways. So they're similar to, but biologically and genetically distinct from, we believe, surface bacteria. Although we haven't sampled lots and lots of these. That's one of the caveats we have to throw out for you today. All these interesting ideas and discoveries, very tiny sample size. We haven't bored down deep more than half a kilometer, very many places in the earth, and the number of pure samples of biological materials from those deep holes also very tiny. So although we believe there's a whole planet full of life down there, we have nothing like a planet-wide, even casual first pass survey. We've done little pinprick studies, various places around the globe. There's, of course, fungi, similar to earthly terrestrial molds and fungi that we're used to and put up with. Animals, we have things like nematodes, flatworms, rotifers, annelids, arthropods. So we have lots of little. If these were in the ocean, we would call them krill, right? These are soil critters for the most part. We have similar critters in soil in your back garden. Uh, the nematodes, the flatworms, the rotifers, they're all there. The arthropods are all there in your back garden. Viruses, these have actually been detected. And so there are viruses that prey upon the bacteria and the archaea, and maybe some of the animals too as well. We don't know. So there's quite a diverse set of flora that live. Excuse me, I suppose fauna is the correct word here, not flora. There's no, there's obviously no photosynthetic plants down deep, and the various times we've had interest in deep life in the earth, it's kind of waxed and waned. And whenever, yes, whenever you discuss topics like this, you're going to get emails like, hollow earth. No, sorry, no. The earth is quite solid. Thank you.
Don't live underneath the earth. It's too hot.
Yes.
You get down that far. It's too hot.
This idea that there was a space, maybe a mile or so beneath the surface, and you see it in fiction like Journey to the Center of the Earth by Jules Verne. The idea that you could go a mile or two down and you could have a temperate environment with phosphorescent plants and things that would give light, and there would be this whole civilization down there. But it's another Atlantis story, isn't it?
Yeah.
Right? No evidence for it at all, sorry, and much to the contrary. Nobody's ever drilled a hole and punched through to empty space yet either, so there's that.
The Japanese have the largest, deepest one, I think, right?
Yeah, there's a big one in Japan. There's some deep mines that are natural spaces in the Americas. The Russians, I think, at one time had what they claimed was the deepest borehole somewhere out in, I don't know, in eastern Russia, somewhere in the wastelands out there. They claim to have the deepest borehole at one time. So who's to say? I don't know. But nobody's gone more than a couple kilometers deep at the most. And most places, 500 meters is quite deep. But you do find some natural mines. You find some of these physics experiments, these neutrino detection experiments, where they get a mine that's a couple kilometers deep, like a lead mine or something like the salt mine that's a couple kilometers deep. And they put experiments down there. But we haven't really explored that much, that deep. One of the things that came up in the 1950s, the U.S. Department of Energy was doing a lot of experiments with their happy new toy, the atomic bomb. Yeah. And the United States, of course, is the most bombed country on Earth because we set off an awful lot of bombs, like over 900 of them, on the continental United States. Hooray! And they were experimenting with nuclear reactors, both breeder reactors for fuel and weapons research, as well as power reactors. And one of the questions is, what do we do with the radioactive waste? Because the fission designs of the 50s through the 70s, not that great, to tell you the truth. They're loads better now. But think of a 1960, early 60s automobile compared to something you would see on a lot today. The technology is wildly different. But they wanted to say, maybe we could bury some of this stuff kilometers deep, like in one of these salt mines, right? We'll buy a salt mine and we'll bury radioactive waste there. And then the subject comes up of, we think there's a whole biosphere down there. So if you're burying your sealed drums of waste somewhere where there's an active biosphere, are the bacteria going to corrode the seals on your drums of material? Are they going to attack the storage materials? And so, all these things, these questions come up, and so ultimately, we didn't take the route of burying a lot of stuff that way, just burying it in drums. Hooray for us, I guess. But there was another thing that caused us to not do studies of the deep biosphere. And the problems of surface contamination and how do we get an uncontaminated sample were discussed from the 1920s on, perhaps earlier than that. But then we had this interesting situation in 1968, the DSV Alvin, and I think that stands for Deep Submersible Vehicle. It's one of these early submarines designed to go thousands of meters down into the sea. And they were getting ready to launch the Alvin. And so the Navy has kind of jury-rigged a catamaran. They have a couple of hulls bolted together so they can lower the Alvin into the sea in the gap between them. And the scientists are in the Alvin. The hatches are open, and this thing is hanging from cables underneath the tender ship Lulu. Lo and behold, the cable snaps, the Alvin plunges into the sea, hatches wide open, and begins to flood and sink. The scientists the three of them manage to escape, but the Alvin sinks with all their gear and the lunch boxes of the three scientists. And so this ship, this Alvin, is lost in 1,500 meters of water. That is not insignificant, friends. You're talking a long distance. That's close to a mile, about 5,000 feet of depth, if you're thinking about that. And so how do we get this sub back? Well, they had another deep submersible vehicle, and they managed after almost a year. This thing was lost in October of 1968, and it wasn't recovered until late August of 69. So it's been down there almost a year. Well, they go ahead and they open the lunch boxes, just wonder what the sandwiches look like, and there's no sign of bacterial decay. Obviously, everything is soggy, but there's no sign of bacterial decay. And so the assumption is, well, obviously, they didn't have any bacterial decay because there's no bacteria down there, right? So the idea is the lunch boxes, the lunches didn't rot because there's no bacteria down there to rot them. And so this just adds on to the critics who, from the earliest days of deep biosphere research, have been chanting, no life, no life, no life. And the reasons are simple. Oh, well, the pressure is too high, the temperature is too high, and there's no oxygen, and there's no free circulation of minerals and biological resources, and if this sounds very much to you like the dry Mars chanting of dry Mars, no water, no life, Mars never had water, oh look, and in spite of all the evidence that continued to be compiled, well into the 90s, there were people saying no water on Mars at all, none, zero. Well, we're looking at this, and people are seeing this lack of bacterial decay in this one circumstance and saying, gee, the whole planet on a deep level must be sterile. And so we come to the question, and we dive into these philosophical issues on this program from now on time to time. The idea of how do we make the change from, oh, we think this is right, to now we toggle over and we think that is right. We change our mind. How do we go from, oh, the Earth is fixed in the center of the solar system, and no, the Earth is a planet orbiting around our sun? How do we make these leaps? And so we come up to the question of, gee, here I have evidence of something that requires a significant change in thought. Is the evidence convincing? And this is what the flat Earth uh, fans struggle with all the time. They look at the evidence presented by people like you and I, saying things like, "Oh, yes, the Earth is curved, and I can, with a stick and a protractor, I can measure the diameter of the Earth." And but no evidence is convincing. So there was a group. John Parks is the lead scientist in this group from the University of Bristol, and they were doing work on seafloor cores. And from 1983 on, these people were saying, "Ooh, look, we have an abundance of cellular life in these deep sea cores." And all of the, deep Earth, dead Earth people were going, "No, no, no, silly boy, there's no life down there. The sandwiches, the lunch boxes of the Alvin, it would have been all rotten if they had bacteria there." And so this John Parks fellow from the University of Bristol is saying, "No, I'm finding between 10 to the fourth and 10 to the eighth cells per gram." For those of you who don't speak scientific notation, it basically means the number of zeros. 10 to the 4 is 10,000. 10 to the 8th is 100 million. So we're talking about a lot of cells. If you're talking about 100 million cells per gram of soil. How big is a gram of soil, friends? Well, I have some clay here. Let's pinch off a piece that may be a gram. So probably a little ball of clay about so big is about a gram. I would have to get out a scale and measure this, but I would say it's pretty close. A gram of soil is quite small, friends. A gram of soil is quite small. And so that means a lot of life. Now, how much is in your garden? If we went out to your backyard and we dug down 5 or 6 inches into the soil beneath your lawn, what would we find? About 10 to the ninth. 10 to the ninth is about 1 trillion cells per gram, or about 10 times more than the richest deep sea sediments. But still, you're talking about, if you're talking about within a power of 10, you're talking about, here's, there's 10 times. You're looking at now, we've got this situation where, my goodness, there's a lot of biology down there. And nobody's been doing, to my knowledge, I couldn't find anything on gene sequencing yet. Well, what do you do when you've got scientific evidence that kind of goes against the current grain? And I get these things every once in a while. Somebody will send me a book or an email or a link to a website. "Oh, here, look at our evidence." And these folks are always very sincere and generally quite decent. But you look at the evidence and it doesn't hold up. And so they say, "Well, why do you not believe mine? And I should believe yours." Well, how come? Well, the prevailing thought was so very much against the idea of a deep biosphere that Parks' group took something like 4 years to get their data actually accepted and published in a scientific journal. People were just rejecting it, saying, "No, no, no, this isn't true. This goes in our file with perpetual motion machines and, flat Earth proofs and things like that." Actually, there were, the skeptics, they said the same things all the time. Gee, basically what it amounted to is the skeptics were saying the environment deep within the Earth is so different from what we experience here. Certainly nothing could live there. That's the core of the environment. Now, for the deep Earth people, the critics said, "Oh, it's too hot. The pressure is too high." Hello, Cynthia. I see you're on our chat today. Appreciate it. Glad to have you here. It's too hot. The pressure's too high. There's no oxygen. There's no. Oh, and Book Davies has joined us. Welcome, friend. There's no reservoir of carbon, right? You say, why a reservoir of carbon? Well, whether you're talking plant or animal, it requires metabolism. Something's got to eat something else. You've got to have a consumable and a waste product that somehow gets recycled in this, or the biosphere dies. You can't be talking about an active biosphere if it doesn't recycle the waste, if it doesn't have a supply essentially of nutrients, of foodstuffs. So basically they're saying, oh, this environment is too terrible. There's not enough foodstuffs. There's no light. There's not enough. Either there's not enough heat or there's too much heat. Depends on who you want to talk to. The pressure's too great. There's no oxygen. This is ridiculous. You can't have life there. And yes. Hey, Book says howdy. Good to see you, my friend. Good to see you joining us. So we're looking at this and we're saying, almost every environment on the Earth, and I'm talking 21st century science here, almost every environment on the Earth that's accessible, there's some kind of life that has colonized that. Some of this life lives in a slow motion dance, a million times slower than you and I live. Their chemical reactions are every few days versus, tens of thousands per hour that power us. These life forms live where stray ultraviolet light carried in by veins of quartz fractures ice and allows it to have access to free water for a few seconds before everything refreezes again. You're talking life that lives in a very, very alien environment. And by 1998, there were lots of little studies. It wasn't just our friend, Mr. Parks, from the University of Bristol. There were lots of little studies, and their group of scientists went ahead and they did what's called a meta-study, where they look at all the data from everything that's been published on this topic to date, and they gathered together 12 years of data and published in 1998, which means they were going back to 85, 86 or thereabouts, and indications from relative abundances of prokaryotic, what we call the archaic life, the Archaea, the relative abundances that you find on the surface versus deep in the earth, the prokaryotes versus the eukaryotes. Eukaryotes are things like me, you, and the grass in your yard and the fungus that grows in the soil there. It all has nuclei in their cell. The prokaryote life, like the Archaea, have no nucleus. They're very primitive. And the meta-study indicates that 95% of the prokaryote life on Earth lives in the deep biosphere, kilometers below the Earth. And you have to wonder, current thinking, some 70% of all the microbes that exist on Earth may live in the deep biosphere. Think about that. 70% of all the bacteria on Earth, we live with a paltry, with the trillions of bacteria that live on every square inch, and our battle with disinfectants to clean them away. You know what? 70% of all that bacterial life may be deep beneath the Earth, which we can't get to, we can't touch, we have no influence upon. That's a lot of life, even if it grows slowly. This is all carbon-based life. Hey, that means we have something like 15 to 25 gigatons of carbon sequestered permanently in the deep subsurface, because there's no ready route of exchange. Now, you think about this. Can we think of environments separated by more than 500 meters? Sure. There's the surface oceans in the Antarctic, and there's the deep sea floor, say 3,500, 4,500 meters below. Well, things in the ocean above die and their remains fall below, and there's a whole biosphere of fungi and bacteria that consume those dead things that rain down upon them. And the nutrients do get recycled because there are currents of water that flow along the bottom of the sea and eventually rise to the surface. And there's this deep current which submerges in the Antarctic and travels along the seafloor and resurfaces in the Arctic. And when it does, it dredges up these nutrients and recycles them from this deep biome, thousands of meters below the sea surface. But there's no comparable exchange mechanism for relatively continuous, large volume, rapid exchange between the deep biosphere and the surface biosphere. What's going to exchange? Do we have vertical rivers that drop miles into the earth, or bubbling springs that rise up from miles deep? Well, the only stuff we have that regularly rises up from the deep interior is volcanic in nature. And so we're talking about, could we have life and biological material dragged up on the fringes of a magma column? You know, the hot water and stuff being dragged upward and ejected from the volcano as steam. Could we get maybe? That's a pretty extreme environment, but maybe some life. But lots of life, lots of nutrients, lots of materials? No. So how do we get the carbon down there in the first place? Tectonics and subduction zones. There are places in the Indian Ocean, off the coast of Japan, off the coast of California, where one tectonic plate slides beneath the others. And Fukushima, the 8.7 or whatever it was quake, and they had the massive subsidence, and that produced a tsunami and all the damage and destruction in Japan at that time. Well, this subsidence also dragged many tons of carbon, both living materials and non-living materials were dragged deeper into the Earth's interior. Now, does life survive the process of being dragged down into one of these subduction zones and into the deep? Do they actually inject live new material into the biosphere down there through this sort of tectonic action? Hard to say. With bacteria, it's a good idea never to say never, I think. But no, certainly no studies on this. But we know this is happening. And so here we have 15 to 25 gigatons of carbon, comparable to the surface carbon mass of all life on Earth currently. Like, wow, that's a lot of stuff. And how much space? Oh, aren't they cramped down there? No, we think they have this is a minimum, a conservative estimate, volume equal to twice that of all of the Earth's oceans combined. So they have plenty of room down there. The other interesting thing, you think about these deep environments, and certainly can you have exchange? Yes. Can that exchange be quick or voluminous? Probably not, but that doesn't mean zero. And when you talk about billions of years of time for life existing on a planet, there's time for a lot of exchange, even if it's really, really slow. So when we're looking at this and we're thinking about these ideas, we're thinking, how does this life get there? Well, one idea is the what some scientists call the oxygen holocaust. There's different names for this event. When photosynthetic life burst out onto the surface of the Earth, and hey, here's a life form that can use radiation directly from the sun. It can fix carbon and create glucose and cellulose and all these other lovely molecules, and it excretes oxygen. Well, up until this time, the Earth's atmosphere, nitrogen, carbon dioxide, methane, it was a reducing atmosphere. Now you have all these organisms excreting oxygen. Well, it doesn't take very long for the oxygen content in the Earth's atmosphere to rise. different things, different changes happened. Fires are now possible. You couldn't have a fire in a reducing environment, nothing would burn. But in an oxidizing environment, you can. Oxygen empowers animals. Animals in an anaerobic environment are very small, very slow, but in an aerobic environment with oxygen to power high speed, high energy metabolic pathways, we can have things which swim, run, crawl, fly, all sorts of things. We can have predator prey relationships are developed that you just don't get in a plants only environment. Well, as the atmosphere becomes more polluted with oxygen, what happens? These archaea, these anaerobes, flee underground, or at least that's the only environment for them that isn't so polluted that it doesn't destroy them. They live in the only place open to them. Are they ported down into the Earth's interior by subduction? Quite possibly. Fluid exchange, draining through areas into the Earth's interior? Quite possibly. But obviously, this aerobic life flourishes there because the kilometers of rock shelter it from the nasty, corrosive, terrible, evil, bad, wrong oxygen that's polluting its nice methane atmosphere. Thank you very much. And then we start speculating, could this be an analog for Mars? If we have a deep biosphere on Earth because the surface became very, very nasty for the early life that was there, could Mars indeed have a deep biosphere? A place where life migrated and thrived when its surface atmosphere, it didn't become contaminated and polluted with terrible, awful oxygen, but rather it dried out. It got very cold. It got irradiated. And it got otherwise, it's just the same way of saying the surface isn't hospitable anymore. Let's go deep. Let's go down inside. And so, could life on Mars have migrated into the interior of the planet? Well, I think it's probably more likely to say, how could it not? And if life on the surface was exterminated, or maybe practically so, then could we still have a flourishing biosphere on Mars? Oh, absolutely. But it's going to take the kind of drilling and archaeology that's going to require boots on the ground. I don't think we're going to see this. I don't think we're going to see like an Oklahoma drill rig operated by robots scurrying around on Martian surface anytime soon. I think we're going to have people there to find that. So it's kind of interesting. But the other thing you have to remember, the surface on the Earth, although we love it dearly, it's very beautiful, nice sunset, sunrise, all the other things we enjoy here, the surface is a really dangerous place. We have megafauna. We have things like alligators and sharks. We have solar storms. We've been getting solar storms in the last couple of weeks. We've been getting coronal mass ejections and large, windy today, so my allergies are going kind of crazy. We've been having lots of solar storms and aurora events much farther away from the poles than we usually have them. You can have severe weather here. We had a tornado touchdown in the town where I live. We had one travel through central Arkansas just a couple days ago. So we can have hurricanes, we can have tornadoes, earthquakes, volcanic eruptions, and these terrific violent events can kill thousands at a go. We can have asteroid impacts. Yes, and it doesn't have to be a very big asteroid. You just have to be too darn close. Keep that in mind. It could be something, it's relatively small. It's only 50 meters across, half the size of a football pitch. Yes, but if you're within a mile and it's a vertical strike, it's going to be a bad day. So, you know what? None of these things affect the deep biosphere much. I suppose Yes, if you say, oh, but hey, Doc, what about 65 million years ago? What about the 10-kilometer rock? What about the Chicxulub impact? Wouldn't that affect the deep environment? Wouldn't that punch through? Yes, it certainly would. And if you were a nematode or an archaea or a bacterium living directly below the Yucatan 65 million years ago, you would have had the proverbial bad day. But you know what? This is like a forest fire compared to the entire biosphere of the surface of the Earth. So, okay, you had an area that was a few hundred square kilometers, and maybe a small core of that might have punched in and injured the deep biosphere. It's like a forest fire. Oh, 10,000 acres have been burned. Is that bad? If you live there, yes, it's terrible. Compared to the entire biosphere of the Earth, is that a terrible event? No, this is one of these things. It happens all the time. Except for the deep environment, it almost never happens. It's protected from almost everything. Okay, if the sun goes red giant, yes, it's going to be a bad day for the deep biosphere. If you have a Theia type impactor, the size of Mars, and it impacts the Earth, yes, that's going to be a bad day for the deep biosphere. But apart from those, literally end of the world type scenarios, the things that we consider to be disasters just don't really touch the deep biosphere. It's an untouched remnant of life as it was on Earth, perhaps 3.5 billion years ago. Does Mars have a similar deep biosphere, Europa, Enceladus, various other moons? Pluto? Pluto, there's thought that Pluto may have a deep ocean. It might not be water based, it might be liquid nitrogen based. Does that mean it can't have life there? Well, we've got a lot of work to do, friends. So we're going to wrap it up here for this week. Paul, did we get any interesting questions on the comment section?
No, they were just really interested in what you had to say today.
Okay. Well, we'll wrap it up there. Folks, you're welcome to email me at astronomyforeducators@gmail.com. It is spring semester, and it is the lousiest, most difficult, and congested time of the year, the busiest time of the year for me as an educator. So if you contact me and I don't get back to you right away, my apologies. I do respond to all my emails, and you will hear from me. It's just sometimes that paying workaday job where I'm responsible to my students to get their work graded and things like that take precedent. What's the deepest life we've found anywhere on Earth? Look, I think that we have found some samples in diamond mines in South Africa where we're talking several kilometers deep. And we have found bacteria, archaea, nematodes, the various flatworms and fungi. And so I don't know. That would be a question for the Google machine, but I would bet it's deeper than 3 kilometers because I know we've got mines that deep. So, and no, as much as I would like to go to Mars, I have no desire whatsoever to go down into a mine. A couple of years ago, I went down into Mammoth Cave out in the southwestern United States. And I'm like, this is why I don't like mining. So there we go. All right. So we'll go ahead and wrap up for today. We hope we'll see you again next week. We'll be back to our regular space exploration agenda. There's lots of interesting things going on Mars and other places. We're also talking about new programs. Question?
Dr. Barth, did you see the new vehicle that I've got?
No.
So I show off. I like to show my stuff off sometimes. I can't help it. There it is up on the screen.
Shiny.
Yeah. Well, it's Bronco Sport, but it's not a full-size Bronco, but I don't need a full-size Bronco. I'm not a rock crawling.
No, that would be just right for Mars. I'm quite sure.
We took that up to the big park, Buffalo National River.
And that's great. That's great 4-wheeling country.
Yeah.
And but you think about a vehicle like that, about that size and weight. Obviously, it would probably have to be an electric propulsion system, but
on Mars, man.
And an atmosphere, an environmentally sealed cabin.
Yeah. Well,
you probably got people in spacesuits, so you want a little bit more cabin room, I'm guessing.
Well, you can only get if you're taking the car with you. There's not going to be much room for people because that's a lot of payload.
Either that or you assemble it there.
3,500 pounds. I mean. Anyhow.
All right. Okay, friends. Thank you all for joining us this week on How Do You Know? We'll be with you next week, as usual, for our Monday deep dive into space exploration and the news of the day in science. Thank you, everybody. Bye-bye now.